main classes of cell surface receptor. We have ion channel couple receptor. We also, the second type, we also have the G protein couple receptor. And the last class of the cell surface receptor is enzyme couple receptor. The actions and also mechanism to induce signal from in the environment into the cells also are different. Channel couple receptor. When a signal molecule binds to the receptor, or the ion channel is ion can go through. We won't learn much about this kind of ion channel receptor on the cell surface, but we will have an example is this kind of receptor on the ER membrane with the consistent later. So the second art is the G protein couple receptor. We will go into detail of this receptor. And the third part, the enzyme couple receptor, we'll also talk in detail in the later part. And as you will also remember in the video what we watched at the beginning of the lecture, the fibroblast, the fibroblast, they also have such kind of receptor. Do you remember the receptor receive a signaling molecule and the second receptor assemble to the first one, and they form a dimer and pop it to bind the signal molecule and transduce the signal into the cells. Yeah. So both of these receptor type we will talk in detail, and I hope that you can remember about how they transduce the signal and what is the mechanism, also the structure of this receptor, and so on. So first of all, the G protein couple receptor. Please remember that, remember the structure of the receptor, and also how it gets activated through the G protein. First of all, let's talk about the receptor. The couple receptor consists of a single polypeptide strand that crosses lipid B layer of the membrane 7 times. Where's my mouse? Okay, it's crossed. It's a polypeptide strand and it's crossed the cell membrane 7 times to form like a cylinder. And it serves as a pocket for the signal molecule. When the signal molecule binds to the receptor, it changes the configurations of the form of the receptor so that signal can be induced. When signal molecule bind to the receptor can interact with the G protein. And the G protein composed of 3 subunits, as you can see here. The alpha, beta, and gamma subunit. Please remember. Within the alpha, beta, gamma subunit, the alpha subunit can bind to GDP or GTP, and its control its activity. When it's bind to GDP, the alpha subunit and also the whole G protein is inactive. On the other hand, when the alpha subunit bind to GTP, the alpha subunit is activated, and also the beta gamma subunit is also activated. It's all about the structure. And now again, what happened in detail when the signal binds to the molecule. You can see here at the beginning in an inactive scenario. When the receptor, in the absence of the signal molecules, and the receptor is inactivated. The alpha, beta, gamma of the subunit of G protein is also inactive because the alpha binds to GDP. When the signal molecule comes and binds to the pocket of the receptor, it induces conformational change, and the structure of the protein intracellularly also change, so that it can interact with the alpha subunit of the G protein. When alpha subunit interact with the receptor, GDP is released. And because in the cells, the concentrations of GTP is high, it's bind automatically to the alpha subunit and activate the alpha subunit of the G protein. The alpha subunit, when it's bind to GDP and get activated, is dissociated from the beta and gamma subunit. And also this beta alpha gamma subunits, they become active. So both of the alpha with GTP and also the beta gamma subunit can activate target genes and induce signals. Here, as you can see, that the active GTP-bound alpha subunit, it can activate or regulate the target genes. And when it activates the target genes, the GTP is hydrolyzed into GDP again, and is from the active form go back to an inactive form. And when the alpha subunit bind to GDP, the alpha subunit become inactive and is associated again with the gamma and beta subunit to form the inactive G protein. So do you have questions here that you have to understand very clearly? And that I will ask you to to explain the mechanism, how the G protein coupled receptor activated and how the activity is regulated. Does somebody have questions here? Hi. Do you have questions?
No.
Okay. So do you understand about the mechanism, how the G-coupled protein become activated and inactivated again?
Yes, teacher.
Okay, so let's continue. So some G protein couple receptor regulate protein channel, ion channel. For example, in the case of acetylcholine, as we said, in the heart pacemaker cells, it reduces heart rate, right? So what happened here? The receptor acetylcholine is a G couple receptor, G protein couple receptor. So when the acetylcholine binds to its receptor, it activates the G protein, as we talked previously, so that the alpha subunit can bind to GTP and or dissociate from the beta and gamma complex, right? And the beta gamma complex can go to the kalium channel and open the channel so that kalium can go kalium can diffuse. And in which directions do you know? Do you know where in the cells they have high kalium concentrations? Do you know, guys? No? Ở đâu mà ở trong tế bào thì có nhiều kali à em? Ở phía trong.
Intracellular.
Kali is present at a higher concentrations in within the cells, and when the kalium channel opens, ions can go from inside the cells to outside. Okay, so what happened with the hot cells? So normally the different ion channel will coordinate to make a heartbeat, and it's happened like this. A heartbeat happened when at the beginning natrium channel open, so natrium come from outside the cells into the cells and make the membrane from negative to positive. And after that is when the membrane of inner cells is positive, it can open other channels so that it make a change in the voltage of the membrane. And each of these, the ion channel, they connect with each other so that the heart beat one beat. So if in case when acetylcholine is present, it activates the G protein and opens the kalium channel. So kalium can come out of the membrane, out of the cells. What happened here is the membrane charge is more go down, so it gets more negative, right? So it takes a longer time for the ion channel to open to achieve a positive voltage. So that is the heart it requires more time for the heart cells to beat, right? So what happened after that? Because all the process of signal conductions is very well regulated. When acetylcholine is present, our heart beat more slowly. But if it's disappear, we need a mechanism so that the system will come back, will normalize, right? So here after the alpha subunit of the G protein is bound to GTP, and so the GTP will be modified, will be will be hydrolyzed into GTP, and when the alpha will reassemble with the beta and gamma subunit again and inactivated. Beta gamma open the kalium channel anymore, and the channel is closed, and the heart cells they function like before. So the first can interact with the ion channel to open or close the channel. The other mechanism to induce signal into the cells is through through two groups of membrane-bound enzyme. The first group is adenine cyclase, and adenine cyclase will transduce signal through a secondary messenger molecule called cyclic AMP or CAM. Here you also may know that ATP is a reservoir of energy in the cells, or the cells save energy in form of ATP or GTP. They also use ATP or a substance produced from ATP as a signaling molecule or secondary signaling molecule is called, so that here you can see that the hydrolyzed 2-phosphate group make a cycle, and that's why they call it cyclic AMP. The cyclic AMP is very unstable. So it can be hydrolyzed very quickly into AMP, and when in the form of AMP, they don't transduce signal anymore. But the cyclic AMP is a very common secondary messenger molecule, and we can learn how it works. Here is some example of signaling pathways that use cyclic AMP to transduce the signal. For example, adrenaline. As we said that adrenaline, when we experience danger, our body secrete adrenaline, and it make our heart beat faster, increase the heart rate, and it contract our skeletal muscle, and it also induce the hydrolysis of fat or oxidations of fat to give our body energy. So you can see here that many processes that we know in our body use cyclic AMP to transduce the signal. Another example is, you can see in this figure, is neurons, and when we add serotonin, is a hormone, it makes us happy. When we add hormone to the neuron, you can see the signal of cyclic AMP, which are present here in yellow and red. The higher is the concentrations of cyclic AMP, the darker is the staining. You can see within 20 seconds, the cyclic AMP signaling was upregulated in the neurons. So let's watch this video. It's also very interesting.
Epinephrine is one of many hormones that is water-soluble, hydrophilic, and therefore unable to cross the hydrophobic plasma membranes of its target cells. Instead, it binds to receptor proteins located in the plasma membrane and does not enter the cell. When epinephrine binds to beta-adrenergic receptors on the liver cell, G proteins on the inner side of the cell membrane are activated. Each G protein is composed of 3 subunits, and the binding of epinephrine to its receptor protein causes one of the G protein subunits to dissociate from the other 2. The G protein subunit which dissociates from the others carries a GDP, which is replaced by GTP when the subunit is activated. The activated G protein subunit then diffuses within the plasma membrane until it encounters adenylyl cyclase, a membrane enzyme that is inactive until it interacts with the G protein subunit. When activated by the G protein subunit, adenylyl cyclase catalyzes the formation of CAMP from ATP. The CAMP formed at the inner surface of the membrane diffuses within the cytoplasm, where it binds to and activates protein kinase A, an enzyme that adds phosphate groups to specific cellular proteins. In liver cells, protein kinase A phosphorylates and thereby activates another enzyme called phosphorylase, which converts glycogen into glucose 6-phosphate. The glucose 6-phosphate is then converted to glucose. Through this multi-step mechanism, epinephrine causes the liver to secrete glucose into the blood during the fight or flight reaction.
So what happened, as you see here in the video, that is what we also talk about as an example of the liver cells. And so please remember very well about the actions of the G couple protein, the G protein couple receptor, and how it activates the alpha subunit of the G protein, and how it transforms the ATP into the cyclic AMP, and after that, the cyclic AMP interact with the phosphokinase A, and the phosphokinase A become activated and trigger the activations of other target protein. And in the case of liver cells, it induces glycogen breakdown. Glycogen is a form that store energy in our body when we need to, you can imagine when we experience stress situations, we need energy for our body to prepare it. So in our fat cells and liver cells, it induces the process of glycogens and breakdown, and also fat oxidations. So here, please remember the signal conductions can happen in both ways. We talk a lot about activations of target, activations of the receptor, and also activations of target genes or target proteins. However, we also should keep in mind that it can also inhibit the receptor, and it can also inhibit enzyme, upstream enzyme. For example, the adrenaline or epinephrine is just a different name from the same stuff. So adrenaline, as we said, is activates the adenine cyclase to produce cyclic AMP from ATP, right? On the other hand, we also have, for example, adenosine is also a G protein couple receptor, is also activates G protein here, but this G protein is an inhibitory G protein, so it inhibits the enzyme adenine cyclase. So if adenosine binds to its receptor, less cyclic AMP will be produced, in contrast to the case of adrenaline, as we told us. Or in the video is glucagon that induce cyclic AMP signaling in liver cells and cause glycogen degradations. So as we said that cyclic AMP, it can induce or activate downstream enzyme, for example, it change the enzyme activity of the metabolism. It can also induce or inhibit genes expression. So cyclic AMP again here can interact and activate the protein kinase A. The protein kinase A will go into the nucleus and interact with its specific factor to induce genes expressions. For example, in our brain, when we learn something new, or when we have to memorize something, cyclic AMP and the signaling through protein kinase A play a very important role. Do you have some any questions about the signaling through cyclic AMP of this part?
Can you re-explain the signaling through cyclic AMP1 for me?
Cyclic AMP1, okay. So here, yeah. So we'll talk a lot about when the G protein couple receptor is activated, it induces the activations, or it activates also the alpha subunit of the G protein in which it binds to GTP so that it activates the adenine cyclase. The enzyme can synthesize cyclic AMP from ATP. I think that you already know. Sorry. The second part of this cyclic AMP have two main mechanisms to affect its target protein and also induce a cellular response. The first mechanism is that it activates the protein kinase A, and the protein kinase A activate other target protein. Here's other enzymes, or it can also inhibit, but here's in this case is activate other enzyme to induce here, for example, in the liver cells or in muscle cells, it induce glycogen's breakdown. Yeah, it changes some enzyme activity. Okay. Do you understand, Minqiao? Here's a protein kinase A phosphorylate.
Yes, I understand it.
Okay, so it's phosphorylate and activate the target enzyme. And here I just want you to know that it's not just one way that activations, but it can also the opposite effect that is inhibit a target enzyme. Yeah. And here is a second mechanism that is the same from the signaling until the activated protein kinase A, but after that, instead of active other enzyme, here is go into the nucleus and is activates the transcription factor and induce gene expressions. Yeah, I think that's clear. Right. It's okay? Should we continue?
Yeah, it's more clearly now. Thank you.
Yeah, just interrupt me if you don't understand something. For me, it's very important that you understand and can follow. It's much more important than if we talk a lot, but after that we cannot keep in mind anything. Okay, so it's about cyclic AMP. So I think just come back here to give you the overview again. Actually, I should. Okay, here. So we talk about adenine cyclase, right? And the adenine cyclase generates cyclic AMP. It's the one mechanism how the G protein can regulate or induce cellular signal. And the cyclic AMPs, they can induce cellular response through two mechanisms, as I explained again recently, to change the enzyme activity or change the gene expressions, right? The other mechanism how G protein can transfer the signal into the cell is through phospholipase C, and it acts from phospholipase C through other secondary messenger protein, secondary messenger molecules such as IP3, deacylglycerol, and cyclic CANC, and that we learned now. So just come back here. So here. So again, the mechanism is very similar when G protein is activated. The beta gamma subunit of the G protein, they can activate phospholipase C. Phospholipase C can cut inositol phospholipid in the membrane. I think when you learned about the lecture about cell membrane, you heard already about inositol phospholipid, right? Do you remember it? Inositol phospholipid? So it's a very important component of the cell membrane and is involved play a very important role in cell signaling. So here again, that the activated phospholipase C, they can cut inositol phospholipid into two parts. The first of all is inositol 1,4,5-triphosphate, or IP3. If you cannot remember the whole name, just remember IP3. And when you learn more about cell signaling, you can remember later the whole name. It's okay for me if you remember IP3. And the other part of inositol phospholipid is deacylglycerol. And both IP3 and deacylglycerol involved in signal messenger molecule. Please remember that. So what does IP3 and deacylglycerol do? What do they do? So IP3 interact with the calcium channel in the ER membrane. The ER membrane, do you know that they store high concentrations of calcium ions? Do you know that?
Yes.
And how the cells can store calcium ion in the ER? They can store the calcium in the endoplasmic reticulum.
I think it is because the activation of the calcium channel, which is transfer the calcium from the outside into the inside of ER lumen.
Again, could you describe that calcium chain ions should be transported from the cytoplasm into the ER, and what kind of protein or what kind of what is responsible for?
I don't remember that.
Pumps. We remember it. We talked before about for example, the kalium and the natrium concentrations is unequal in the two sides of the cells membrane, right? And it's also the same here with calcium. Calcium are present concentrated in the ER, but in the cytoplasm, the cells the calcium concentration very, very low. It's because they have active pumps that can pump the calcium from the cytoplasm into the ER. Okay, Thao. Thank you. So this the cells keep the cytoplasmic concentrations of calcium very low because calcium is a very important secondary messenger molecule. So in this figure, we have three secondary messenger molecules. Please remember, we have calcium, we have IP3, and we have deacylglycerol. Yeah. So how do these secondary messenger molecules they play with each other to induce a signal? So we talk about the IP3s, they open the calcium channel in the ER membrane, and so that calcium can go from the ER lumen to the cytoplasm and increase the calcium concentrations in the cytoplasm. Calcium can and cancer ion and deoxyglycerol can interact with the protein kinase C, and the protein kinase C, I think protein kinase C can activate its target protein, and it can also change activity as enzyme, and it can also, later we can see, change the genes expressions. On the, in addition, cancer, as I said, is very important secondary molecule, and the cells can sense the cytoplasmic cancer very quickly due to comedulin. Comedulin is a protein that can interact with its target genes in the presence of cancer. Comedulin in the 4 positions as shown here. And the cancer comedulin complex phosphorylate its target genes. And for example, when we learn something and when we memory stuff, cancer comedulin complex present highly at the synapse, and is very important for our learning process. Other example of cancer signaling is shown here. You know that when an egg is fertilized by sperm, only one sperm can fertilize the egg. And do you know what is the mechanism for that? Do you know the mechanism? How, why this, an egg can receive just one sperm but not many?
Theo con nhớ là hình như là khi mà con tinh trùng nó đi vào trong quả trứng thì quả trứng nó sẽ tạo ra cái màng gì để kiểu nó chặn các con khác mà đi vào đấy.
Đúng rồi. Đúng là hồi phổ thông là cũng chỉ biết là ok, nó sẽ tức là thay đổi cái màng của trứng khiến cho các con tinh trùng khác không vào được, đúng không? Nếu ở đây thì chúng ta sẽ học sâu hơn một chút nhé. Thì nếu mà ở đây trong cái quả trứng này thì người ta biến đổi để người ta có thể, tức là người ta có thể theo dõi cái quá trình tức là người ta có thể theo dõi được cái nồng độ canxi ở trong quả trứng này bằng biểu hiện màu đấy, cái màu vàng với cả màu đỏ ấy, càng màu đỏ thì sẽ là càng nồng độ càng cao, sau tiếp đó là màu vàng, sau đó màu xanh lá cây, và ở cái màu xanh da trời là gần như là không có. Thì người ta thấy là chỉ trong vòng 40 giây, tức là ngay sau khi mà tinh trùng thụ tinh vào trứng thì bắt đầu có xuất hiện đây này, sau 10 giây thì bắt đầu xuất hiện ở trong trứng thì xuất hiện cái tín hiệu của canxi, và sau đấy thì cái tín hiệu cứ lan tỏa dần đến hết quả trứng. Và cái như chúng ta hôm nay học thì canxi là tham gia vào quá trình truyền tín hiệu, đúng không? Thì khi mà quả trứng nhận được cái tín hiệu này thì nó sẽ biết là nó sẽ phải, tức là canxi lúc đấy thì nó sẽ bám vào comedulin và canxi, cái tổ hợp này nó sẽ làm thay đổi tính chất của các cái protein đích, và cái protein đích này nó sẽ làm thay đổi cái tính chất của quả trứng. Ví dụ như nó làm thay đổi cái cấu trúc màng tế bào, khiến cho nó cứng hơn, và nó không còn, tức là lúc đấy thì các tinh trùng khác thì không thể xâm nhập vào trứng nữa, được không? Thì bọn em thấy là tức là những cái gì mà chúng ta học thực sự nhé, nó rất là, đúng là nó xảy ra, tức là nó cũng khá là, nó để có thể giải thích những cái hiện tượng rất là quen thuộc với chúng ta. Và cái mà khi mà chúng ta học kết nối các cái tức là liên tục là coi như là học giữa lý thuyết và những cái mà gọi giải thích cho những cái hiện tượng xung quanh chúng ta ấy, thì nó cũng khá là thú vị. So now come back to our lecture again in English. So and calcium also play important role in our nerve cells, in which is the calcium channel at the synapse. When the calcium channel open at the synapse, calcium from outside the cells going stream into the cells, increases calcium signal inside the cells, inside at the synaptic, and induce the infusions of the intracellular vesicle and secrete of the stuff into the synapse. For example, acetylcholine or glutamine. So when the cells, the neuron, the synapse of the neuron activate with calcium signal, is going to secrete acetylcholine into the synapse. So we learned about the signaling conductions through G protein couple receptor, through the ends when exchange the activity of the G protein, and G protein activate and inhibit subsequent enzyme like adenine cyclase or PCK. So it induces cyclic AMP and cyclic or calcium in the cytoplasm, and cyclic AMP or CanC can induce signal into the cells, and here we have to know that these signal conductions can be integrated to give the cells a unique cell response. For example, in this case, muscle cells, neurostimulations induce increase of CanC ion intracellularly, and it's going to activate the glycogen's degradations. So glycogens could be broken down into glucose to give the cells energy. On the other hand, or in additions, actually, hormonal stimulations induce the synthesis of cyclic AMP. Cyclic AMP activate protein kinase A. Protein kinase A also go into these directions to stimulate the glycogen's hydrolysis. On the other hand, it also inhibits the process of glycogen synthesis, and it really makes sense because when the muscle cells they need energy, so they need so they stimulate the glycogen's or degradations, and the cells need to inhibit the opposite process in which glycogen is synthesized, right? So the signal in the so the cells can receive many signals, and the signal will be integrated and coordinate with each other to give a unique cell response. Or in the liver cells, for example, hormonal stimulations, it also stimulates the G-couple protein, and as a consequence, cyclic AMP is formed. And also the deacylglycerol and the IP3, all of them are secondary messenger molecule, and IP3 opened the CanC ions channel in the ER, increase the concentrations in the cytoplasm, as we talked about. And so the signal integrate with each other, and what happened here, deacylglycerol activate together with calcium protein kinase C, and protein kinase C inhibit the glycogen synthesis, right? And on the in addition to the actions through protein kinase C, CanC itself also interact with comedulin, and the comedulin CanC complex stimulate the glycogen degradations in these directions, right? So it's also it's similar to the muscle cells. It makes sense that once the glycogen's degradations take place, that means the cells need more glucose. The synthesis of glycogens should be stopped. And here we also see that cyclic AMP, it activate protein kinase A. Protein kinase A stimulate also like, for example, similar to the Kanzi's process of glycogen degradations and inhibit the upside functions, right? So the message here is that different messenger molecules, they can coordinate with each other to give a unique cell response. So Kanzi signal signaling induce many biological or regulate many biological processes. As we talked previously about acetylcholine and acetylcholine, so transduce signal through calcium through Kanzi ions. So, for example, here as we talk, acetylcholine affects the pancreas to secrete digestive enzyme, or in the salivary gland cell to secrete the amylase, or to induce contractions in the stomach, smooth muscle, or in the liver cells, it induces glycogen's breakdown into glucose. And that you can see that, okay, how all the stuff that we're learning here now is come together. Yeah. Do you have questions here? Does somebody have questions here?
No, teacher.
So I think I try to give you questions. So here's our overview. We talked about G protein, right? How it's become inactivated again. We didn't talk about how cyclic AMP. Actually, we talked about how cyclic AMP get inactivated. Do you guess how? Do you know how can cyclic AMP become inactivated? Do you remember it?
No.
So do you remember the slide, how it's formed from cyclic AMP is synthesized from
ATP.
Right. And adenine cyclase stimulates this synthesis of cyclic AMP from ATP. And do you remember when, so cyclic AMP, I told you that cyclic AMP is very unstable. It will be hydrolyzed into
AMP.
AMP, right. So in form of AMP, the signaling will be stopped, right? It's the mechanism how cyclic AMP is deactivated. Yeah? Understand?
Maybe.
Maybe, okay.
Can you repeat again?
No.
Not sure.
Cyclic AMP is very unstable. It will be how can I say is that you can, so it will form spontaneously into AMP. Yeah. So signal transduced by cyclic AMP is quite short. tức là cái mạch AMP vòng ấy nó rất là dễ, tức là nó rất là kém bền và nó tự động biến đổi thành cái thành AMP. Tức là một khi mà cái tín hiệu từ G protein ấy không còn nữa ấy, hay là cái adenine cyclase ấy, nó lúc đấy nó bất hoạt thì nó sẽ không tạo ra các cái mạch AMP vòng mới, cái phần tử AMP vòng mới, thì lúc đấy tự động là cái tín hiệu nó sẽ bị đứt, được không? Các bạn hiểu không?
Dạ có ạ.
Okay. Now we come to the second mechanism of how G protein induce a signal. Here you can see that G protein activate phospholipase C cut inositol phospholipid into two secondary messenger molecule, deacylglycerol and IP3. IP3 opens the calcium channel in ER and release calcium, and calcium ion and deacylglycerol activate protein Kc. And protein Kc will regulate its target genes. So my question is, how can this signaling be regulated? How can it be stopped? Các em biết, thế tương tự như AMP vòng thì ở đây thì làm nào để coi như cái tín hiệu này dừng lại?
Nó phosphor hóa.
Cái này thì là nó phosphor hóa. Đầu tiên chắc là dễ nhất là canxi em ạ. Canxi thì thế nào?
Thì nó sẽ ngừng
Thì đầu tiên là cái canxi ấy, nếu mà muốn dừng cái tín hiệu canxi này thì chỉ có cách là cái kênh ion canxi này bị đóng lại đầu tiên đã, đúng không? Sau đó nó phải vận chuyển canxi từ ngoài tế bào chất vào bên trong mạng lưới nội chất, đúng không? Mà lúc nãy chúng ta nói là nó vận canxi vận chuyển từ tế bào chất vào mạng lưới nội chất thì nhờ các cái protein, tức là các cái bơm, đúng không? Bơm đặc hiệu, được không nào? Rồi, deacylglycerol với cả EPI3 thì cũng không phải là bền lắm, và chính nhờ thế thì nó cũng, cái tín hiệu này nó được truyền khi mà cái thụ thể nó được nhận tín hiệu nhá. Còn cái protein kinase thì sau đấy thì nó cũng có thể phosphoryl hóa, hoạt hóa bất hoạt, và sau đấy nó có thể có những tức là cái đấy thì tí nữa chúng ta sẽ học sau. Nó có thể hoạt hóa các cái protein mà bản thân các cái protein ấy quay lại để bất hoạt cái protein trước đấy để chấm dứt con đường truyền tín hiệu, được không nào? So now we come to the third group of receptors, the enzyme couple receptor. I think I should come back a few slides to give you an overview where we are, what we have learned. Here. Do you remember this slide? We talk about the three different cell surface receptor. We have talked very in detail about G protein couple receptor, right? This case. Do you remember the alpha subunit of G protein and is activated through the signal and receptor complex, and then is activates the target protein or enzyme, right?
Yes.
Okay. Now we come to the third group of receptor, enzyme couple receptor. and we will watch again the video that we watched at the beginning of our lecture. But before that, we should take a break, okay? Let's say, yeah, we will come back in 5 minutes, okay? See you then. Hey, do you come back? Are you here, Lady?
Yes, we're here.
So now let's talk about the enzyme couple receptor. Third major group of receptor, and it play a very important role, for example, for the cells to receive external signal like such as growth molecule or cytokines for the immune system. I don't know what I should. Okay. So, for example, in case of growth factor and cytokines, the receptor mostly of the time are the enzyme couple receptor, like this case. Why people call it enzyme couple receptor? If you remember about the G protein couple receptor, you remember when the protein receptor is activated or is when it's bind to the ligand, the receptor interact and activate the G protein. And here is a similar thing. When the ligand bind to its receptor, a part of the receptor is an enzyme itself. It can phosphorylate and activate other receptor, or it can bind to an enzyme and activate the receptor or activate the other target protein. So similar to the G protein, please remember very well the structure of the enzyme coupled receptor. Receptor is a transmembrane protein which consists of three domains: the extracellular domain, the transmembrane domain, and an intracellular membrane. Membrane functions as and the transmembrane domain is to keep in on the cell surface, go through the intracellular domain is important to transduce signal into the cells. So how the enzyme coupled receptor become activated? So we have signal, as I said, growth factor or cytokines. The growth factor bind to its receptor on the cell surface. It can induce cellular response as we talk with the G protein. It can induce two different things, or many cell activity. But as we talked at the beginning, that the actual cellular response can happen very quickly. For example, in case of insulin, when the insulin is there, and the cells will uptake sugar immediately. Or in case of in the video, as you remember, when growth factor is secreted from platelet, trong tiểu cầu, fibroblast, tế bào sợi, will receive the signal of the growth factor and is change the shape and the movement. All right, and so it's happened in this case. The cytoskeleton was modified, the fibroblast were modified, in which the microfilament and tubule was changed. So change in their shape and also in their migrations ability. So in our case, when we have an injury, fibroblasts they should migrate to the injury site and repair the cells and tissues there. That's why it's important for them to change patient's behavior. On the other hand, what we will see also that the growth factor, when it activate is the receptor, it also induce genes expressions and triggers the cells in cells, and the cells will proliferate, and so that they can replace the injured cells in the wound. So when genes expressions take place, it happen more slowly. It can take place after hours. We can observe the effect after hours or maybe overnight. While patients cytoskeleton happen within minutes, so it's very quick. So now come back to our receptor. We have two major types of receptor for the enzyme coupled receptor, which is receptor serine threonine kinase and receptor tyrosine kinase. Today we will talk more about talk in detail about the receptor tyrosine kinase, because it's very common, and we use the example with the growth factor and cytokines, so we concentrated more on the receptor tyrosine kinase. The difference is just an explanation. If you can remember, it's good. The first case, the receptor serine threonine kinase, when the receptor activated, the kinase will phosphorylate in the serine and threonine residue of the receptor. And in the second case, the kinase will phosphorylate in the tyrosine side of the receptor. Cái này bọn em có hiểu, tức là cái amino acid ấy thì nó serine này, amino acid cấu phần thì cái cả serine threonine này hay là tyrosine nó đều có thể, khi mà nó phosphorin hóa thì nó có thể là làm thay đổi tính chất của cái enzyme, thì nó có thể là hoạt hóa hoặc bất hoạt, nhưng mà nói chung là thay đổi tính chất và nó tùy theo cái vị trí nó phosphorin hóa, nên là nó chia cái thụ thể này thành hai loại, một là serine threonine, hai là tyrosine. Được chưa? So now come back to our video again. We can watch from minute 3.
Shown in purple encounters a matching receptor.
Đây nhá. Thế so let's watch the video again about the child. She got a wound and what happened inside the cells. Okay, here's a fibroblast. It's a membrane of fibroblast. You see here the receptor. Look what happened when the ligand comes. Okay.
In and out. The gray structures sticking out of the cell membrane are receptors for incoming signals. When the growth factor from the platelet, shown in purple and blue, encounters a matching receptor, it binds to it. A second receptor protein joins in, making the growth factor fit like a key in a lock. Growth causes the receptor to change shape. This change in the protein conducts the signal through the membrane and into the cell's interior, the cytoplasm. You'll see this better from inside the cell. Beneath the cell membrane, you see the gray receptor ends surrounded by pink fibers. These structures help to give the cell its shape and a range of messenger proteins that will carry the signal through the cytoplasm. While active, as shown by the yellow flashes of light, the ends of the receptor interact with the messenger proteins. We'll watch the action again from our position in the cell's cytoplasm. The growth factor binds the receptor proteins outside the cell, drawing the receptor ends together. The signal is transmitted through the cell membrane, and each new protein is activated in turn. If you look closely, you can see the proteins change shape as they become activated with the signal. Each step of a pathway is under tight control to ensure that the correct message is relayed. For example, as this white protein accepts the signal, the blue protein comes in to deactivate the red one. Let's follow the white protein on its journey through the cytoplasm, the center of the cell. As we follow the signal to the nucleus, you'll see it passed from messenger to messenger. The first exchange is with this brown protein. In a few seconds' time. Although we're only following one path, a single cell has many different ways to transmit signals through the cytoplasm. Shown in green are part of the cytoskeleton. Like the pink fibers you saw before, these give the cell shape and help to organize its contents. Crawling along the fibers are motor proteins that reshape the cytoskeleton and help this fibroblast cell to move. On our way, we will encounter other structures in the cytoplasm, known as organelles. On the right, you see glowing organelles called mitochondria, which generate energy for the cell. The activated protein passes by a network of membranes, here in light brown, known as the endoplasmic reticulum. The protein is transported into the nucleus through a pore in the nuclear membrane. The nucleus contains tightly wound coils of DNA, shown in green. The protein messenger passes the signal to two other molecules that team up to locate a specific gene along the DNA. In this case, the gene carries the information to make a growth factor. Other molecules then unwind a small section of the DNA molecule and allow an enzyme called RNA polymerase, shown in brown, to make an RNA copy of the gene. The copy, called messenger RNA, here in light green, carrier proteins and leaves the nucleus.
And after that, a growth factor will be synthesized, okay, from the messenger RNA into protein. Yeah. So you can see clearly what happened with the ligand, with the growth factor when it binds to the receptor, and it changes the shape of the receptor so the signal can induce into the cells.
We'll use this copy.
So here again, in these animations, you can see that our tyrosine kinase receptor tyrosine kinase is in a single form. When the ligand binds to the receptor, it can form together with another receptor to form a dimer. And the signal, the binding is very specific by this receptor and transduce signal specifically to the cells which express this receptor. And what you can see here, so that after the cells, the signal binds to the receptor like a key into the lock, the enzyme part of the receptor, tyrosine kinase, they come so close together that they can phosphorylate and activate each other. So here you can see that they phosphorylate in different positions, and this position, so here they phosphorylate the tyrosine size. That's why they call that the receptor tyrosine kinase. The phosphorylated tyrosine lock other proteins to the activated receptor, and this protein can activate each other and transduce signal to its target proteins downstream. So today that is here is the principle how the receptor tyrosine kinase, or similarly the receptor serine threonine kinase, and to in detail into the downstream process. It's shown here, for example, because we talk about hormone or cytokines or growth factor. They use this kind of signal transduction pathway very often. So later, later you will, if you guys studies cancer, or you go to so you study botanics, but with focus of signal transductions and so on, then you will go very in detail in this pathway. Today I just want to give you an overview. However, what I noticed, because I also teach master students, and I noticed that somehow they hadn't learned much about cell signaling, although it's very important part. 1 second, sorry. Đây, sorry. Em đưa chị Hậu luôn nhé. So, although it's very important in cell biology and also in molecular biology or biochemistry, people study a lot about cell signaling. Many drugs they have developed for inhibitor, or somehow they target in the cell signaling pathway, and so that's why I would like to give you a more close introductions. At least you have heard about some important kinase that come over again in many systems, from, let's say, from fish to Drosophila to to xenopus and also in human and so on. So first, the first system is cytokines, which activates the JAK and START kinase, JAK-START signaling pathway. So we talk about the immune cells. We talk about hematopoietic stem cells today, and we also know that so in the blood system we have 3 major lineage of the cells. That's the red blood cells, white blood cells, and platelet. During the maturations of red blood cells, we need a cytokines called APO or erythropoietin. Erythropoietin is very important to induce red cells differentiations. And how is work erythropoietin? act through the Jack-Start signaling pathway. Here again, when it's bind to its receptor on the membrane, the two receptors can resemble together to activate each other in the cytoplasmic domain. Here you can see an enzyme called JAK. It's a tyrosine kinase. It's bind to the intracellular membrane, a domain of the APO receptor. So it can phosphorylate the receptor at each other and phosphorylate the cytoplasmic domain of the receptor is shown here. After that, the phosphorylated tyrosine will recruit start protein to the receptor so that JAK can phosphorylate the start molecule. Two start molecule will form a dimer. This start dimer go into the nucleus and bind to a specific sequence and triggers the gene's expressions. So it's quite easy to understand. Do you have questions here? Or maybe we can, so first of all, do you have questions?
No, teacher.
Somebody. Okay, so other cytokines is just an example for erythropoietin. Other cytokines which also, for example, inflammatory cytokines. For example, the cytokines in the questions we got previously when we have an infections or mình bị lở mồm hay herpes cũng thế, thì lúc đấy thì cái kiểu các cái hạch bạch huyết ở cổ ấy, đúng không, thường nó hay sưng lên và cảm thấy đau, đúng không nào? Thì tức là lúc đấy thì các cái cytokines viêm được các cái tế bào ví dụ như granulocytes, monocytes, macrophage nó tiết ra và nó làm khiến cho kiểu sưng tấy đỏ và thậm chí là kể cả khi mà bị dị ứng ấy, cảm thấy ngứa ấy, thì cũng là do các cái cytokines này, và cái con đường nó tác động thì là qua receptor tyrosine kinase, và đấy là cái ví dụ của cái Jack-Start này, được không nào?
Vâng ạ.
So if you don't have questions, I would like to ask you, for example, in the Jack-Start signaling, what is the kinase? What is a tyrosine kinase? Cái gì đóng vai trò là cái tyrosine kinase đấy hả các em? Các bạn.
Nó là cái chắc đúng không? Chắc.
Xác. Đấy, chính xác nhá. Bọn em tức là cố gắng học những cái gì gọi là nguyên tắc chung, sau đó thì bọn em cứ đào xem là ok, thế coi như là mỗi cái bởi vì nếu mà các em để ý, thực ra nó có cái mạch học. Nếu mà nhớ nhá, chúng ta sẽ đi lúc đấy là đi từ các cái loại receptor này, xong rồi các loại G protein couple receptor, có các phân loại ở dưới này, xong rồi có các cái secondary messenger molecule này, thì ở đây cũng thế thôi. Ở đây là cái enzyme couple molecule, thì cái nó đều có tại sao chia như thế, bởi vì nó có cấu trúc tương tự nhau và nó hoạt động cũng có những cái nguyên tắc hoạt động tương tự nhau. Và lúc đấy thì bạn em bắt đầu xem lại, kiểm tra lại, ok, thế thì những cái ví dụ này thì cái nào đóng vai trò gì, được không nào? Thế còn start ở đây thì đóng vai trò gì? Các bạn, start ở đây đóng vai trò gì?
Ở đây nó ghi là transcription factor thì nó sẽ là kiểu yếu tố điều kiện để tạo ra việc phiên mã à?
Gọi là yếu tố phiên mã đúng không?
Thì nó chỉ là tên nó đấy ạ.
Chắc là chỉ là tên của nó. Em nói yếu tố phiên mã là mọi người đều hiểu, nói transcription factor là mọi người đều hiểu, nó không phải là chỉ tên của nó. Tên của nó là start. Nó còn có nhiều loại start không? Nó có start 2, start 5, start khá là nhiều. Các cái protein nó có cái cấu trúc tương tự nhau và nó có cơ chế hoạt động tương tự nhau. Ví dụ ở đây là nó đều phosphorin hóa bởi YAP này. Nó được phosphorin hóa tại vị trí tyrosine này. Chính vì thế nên tức là vì cái JAK nó có khả năng phosphorin hóa ở vị trí tyrosine nên nó gọi là tyrosine kinase. Tất cả các protein start này thì nó đều hoạt động theo kiểu sau khi nó được JAK phosphorin hóa ở vị trí tyrosine thì nó có thể tạo thành dimer, và tại khi mà với cái cấu trúc dimer này tức là nó mới đi được vào nhân và nó để kích hoạt cái quá trình phiên mã, được không nào? Nên là nó gọi là yếu tố phiên mã. Đồng thời nó cũng đóng vai trò là cái chất để gọi là như kiểu cái messenger ấy, signaling molecule ấy, để chuyển cái tín hiệu từ thụ thể vào trong nhân, được không nào? Bởi vì rõ ràng như tất cả các cái protein mà gắn với thụ thể, các em thể để ý xem nó ở trên màng tế bào, nó không thể nào mà nó chuyển tín hiệu tiếp theo nếu như nó gắn chặt vào đấy, nên là nó sẽ phải có những cái cấu trúc protein nó khi mà nó được phosphorin, nó được hoạt hóa thì nó tiếp tục nó sẽ có những cái biến đổi để nó tiếp tục đưa tín hiệu nó vào bên trong, đúng không nào các bạn?
Dạ vâng ạ.
Rồi, bọn em có hiểu kinase không? Bọn em biết khái niệm của kinase không? Tại vì cô cứ thao thao bất tuyệt từ nãy đến giờ là kinase, nhưng mà các bạn có biết kinase là gì không?
Dạ em không biết ạ, em chỉ biết nó là kinase như cô bảo.
Nếu mà thế này nhá, không vấn đề gì cả. Cô quên mất không nói trước, theo kiểu có lớp các bạn biết đấy, có lớp như sinh chuẩn trước cô hỏi sao nhỉ? À không, lớp chắc là lớp tài năng, cô quên mất rồi. Thì các bạn biết có lớp biết và có lớp không cũng không vấn đề gì cả, bọn em cứ nghe các khái niệm đi, cứ gọi tên nó rồi đến một lúc nào đấy bọn em có thể hiểu hơn một chút cái khái niệm tổng quan hơn một chút, hoặc là đi từ khái niệm tổng quan xuống cái cụ thể tùy, nó không vấn đề gì, đấy là cái quá trình học. Kinase nhá, nói thế này để cho các em dễ nhớ này. Kinase là các cái enzyme mà nó có khả năng phosphorin hóa các protein khác, được không? Người ta đều gọi chung là kinase, được không nào? Thế thì bây giờ khá là make sense rồi nhá. Các mình đang học là tyrosine, đợi có thể thụ thể hoạt hóa, thụ thể tyrosine, bây giờ chỉ nói tiếng Anh, receptor tyrosine kinase, thụ thể tyrosine kinase, đúng không? Hả các bạn? Thì tức là những cái thụ thể này nó có thể phosphorin hóa các protein khác ở vị trí tyrosine, nên là người ta mới gọi là receptor tyrosine kinase, được không nào? Ở trong cái G protein couple receptor ấy, thụ thể gắn với protein G ấy, các bạn có nhớ là mình có protein kinase A không? Và protein kinase C. Protein kinase A thì được hoạt hóa bởi cái cyclic AMP, nhớ không? AMP vòng. Còn cái protein kinase C thì được hoạt hóa bởi deacylglycerol và canxi, nhớ không các bạn? Thì ở đấy kinase bởi vì nó có thể phosphorin hóa các cái protein đích, được không nào? Được không các bạn?
Dạ vâng ạ.
Thế nhá, thì nhớ cái khái niệm kinase này nó sẽ, nếu mà học về cell signaling, nếu mà bọn em nhớ được cái khái niệm kinase này nó giúp em rất là nhiều. Thế ngược lại nhá, thế cô luôn cũng muốn nói truyền tải với em cái nguyên tắc này, nó đã được hoạt hóa thì nó sẽ có thể bất hoạt, cũng như là tức là nó đã có gắn một gốc phosphorin rồi thì nó sẽ có cái quá trình để gỡ cái gốc phosphat đấy ra, đúng không nào? Bởi vì cái quá trình tế bào thì thường là các cái quá trình đấy nó có thể đảo ngược cho nhau, nên nó gọi là reverse reactions ấy, đúng không nào? Thì nó có cái khi mà kinase nó hoạt hóa, tức là nó xúc tác cho cái quá trình phosphorin hóa thì sẽ có enzyme khác mà nó gỡ bỏ cái gốc phosphat ra khỏi protein đích, được không nào các bạn? Và cái protein đấy nó hay là cái enzyme đấy thì người ta gọi là phosphatase, được không? Nhá.
Vâng ạ.
Nếu các bạn không nhớ thôi để cô ghi vào đây. Kinase. Cô ghi vào cửa sổ chat nhá. Rồi. Và các bạn nhớ cho cô một cái này nữa, gọi là tí phosphatase. Tóm lại là kinase và phosphatase thì có vai trò ngược nhau. Và tức là cái quá trình này, tức là cái quá trình phosphorin hóa và tức là gọi là phosphorylations and dephosphorylations can be reversed, hoặc là đảo chiều. Cái cô lúc nãy cô đang nghĩ là nhá, phosphorin hóa không đồng nghĩa, và không phải lúc nào cũng đồng nghĩa với việc là hoạt hóa protein đâu nhé các bạn nhé. Lưu ý cho cô một phần đấy thôi. Cũng giống như cái ví dụ về cái adenosylase ý, nó có những cái phối tử và receptor nó hoạt hóa cái adenosylase để nó tạo thành AMP vòng. Ngược lại nó cũng có những phối tử và receptor đặc hiệu để nó chặn cái quá trình tạo AMP vòng. Thì tương tự như thế, phosphorin hóa nó lại còn tùy vào vị trí phosphorin hóa cơ. Có vị trí phosphorin hóa của một cái protein thì nó hoạt hóa, và có những vị trí mà nó bất hoạt. Tuy nhiên, tức bây giờ bạn không phải nhớ quá nhiều, nhưng mà tức là cô chỉ muốn là các bạn nhìn nó tổng thể một chút, và cô cũng rất là hạn chế kiểu đúng là như cái này thì đang vì là đưa ra các ví dụ cho các bạn, nên là cô sợ là để tạo thành một cái ấn tượng là lúc nào phosphorin hóa cũng là hoạt hóa nhé, được không nào? Bởi vì thật sự là tế bào của chúng ta nó là một cái thể rất là thống nhất, và nó rất nhiều quá trình cùng diễn ra, nó diễn ra rất đa dạng, và chúng ta đang cố gắng bóc tách để giới thiệu cho các bạn về cái gọi là cách phản ứng của cơ thể với môi trường, của các tế bào với môi trường xung quanh. Nó một mặt thì nó rất là phức tạp, bởi vì nó có sự tham gia của rất nhiều protein, nó rất là nhiều đa dạng, nhưng mà rõ ràng các bạn thấy là cơ thể chúng ta phải đáp ứng rất nhiều thứ cùng một lúc. Bây giờ nhá, các bạn vừa nghe này, vừa xem slide này, các bạn vừa gọi là hít thở không khí này, các bạn vừa biết được là xung quanh về điều kiện ánh sáng, về nhiệt độ, tức là các bạn cảm nhận thấy qua tất cả 5 giác quan. Rõ ràng cơ thể chúng ta nhận được rất nhiều các tín hiệu, cũng như giống như các tế bào thôi, nó sẽ nhận được rất nhiều các tín hiệu. Chính vì thế nó sẽ phải xử lý rất tốt để có thể giúp cho nó đảm bảo được các chức năng của nó, và lại còn đáp ứng được những sự thay đổi của môi trường. Vì vậy nên là không có gì ngạc nhiên là cơ thể chúng ta tức là các cái khi mà cái quá trình truyền tin nó lại diễn ra một cách phức tạp như vậy, nhưng đấy cũng là cái điều kiện tiên quyết để đảm bảo cho tế bào hoạt động bình thường nhé. So let's talk about the next signaling pathway, which is also very common. Is about the RAS MAP kinase. So RAS is a small GTP binding protein. It's also very important to remember. We talk about the G protein, yeah, and the subunit, the alpha subunit of the G protein, they bind to GTP, and is a switch or is regulates the switch on off of the G protein, right? So RAS is similar to the alpha subunit of the G protein. RAS actually is a small G protein. It can also bind to GTP or GDP. When it's bind to when RAS bind to the GDP is inactive. When it's bind to GDTP is activated. Okay. So how can RAS be activated? It happens through the receptor tyrosine kinase. So when a signaling molecule bind to its receptor, the two receptors come close together. It can phosphorylate each other. So the phosphorylated tyrosine residue recruit other adapter protein and recruit the RAS GTP exchange factor to close to the membrane, and it can exchange the GDP bound to inactive RAS with GTP. So RAS protein is now bound with bound to GTP and it's become active. And it can induce signaling into the cells further. So you can see guanine. You can see here this enzyme here which activate RAS protein is a guanine nucleotide exchange factor, and is functions similarly like the alpha subunit of the G protein. And please remember RAS when it's bind to GTP is become activated. Okay. And what happened with the activated RAS? So here you can see again, it can activate other downstream protein. Here is the MAP kinase cascade. Here is composed of 3 MAP kinase. Is the MAP kinase activate with the MAP kinase, and the MAP kinase activates the MAP kinase. You don't have to remember it so in detail, but just remember activated RAS activates the MAP kinase cascade. And as a consequence, it's phosphorylate and change the behavior of many proteins. So the protein activity can be modified, or it can change in genes expressions. So now let's talk about the regulatory mechanism of the protein G and system, and also the small protein G, the RAS. Here, as I told you in the previous slide, is the mechanism how RAS is regulated. You remember this. Here you can see the RAS molecule. Here is inactive because it's bound to GDP. When the receptor is activated and the tyrosine kinase recruit GAF, so guanine exchange factor, guanine nucleotide exchange factor, to close to RAS is exchange GDP with GTP, so RAS become activated, and it can activate the MAP kinase cascade. After that, another enzyme will come into play. The GTP, the GAP is short form of GTPase activating protein, will come and hydrolyze the GTP into GDP and RAS become inactivated again. So you see here's there's two important enzyme that play here a role. This is the GAF and the GAP. GAF is responsible for to activate RAS, and GAP hydrolyze GTP into GDP and making RAS inactive again. Do you have questions here?
Sorry, teacher.
Mm-hmm.
I regret to say, but today we will have the midterm exam at 1 p.m.
Mm-hmm.
So could you let us to have a break on time because we prepare exam.
We can finish our lecture in time, so it's at 12 o'clock, or do you need some more time to prepare? I can stop earlier at 10 to 12 for you to prepare for the exam better, and we will continue this lecture next week, and then we go to the new lecture after that. It's okay for you guys, or what do you prefer actually? For me, it's fine.
That would be great.
Okay, very good. So I finish this slide, and then we have a break. Yeah.
Okay.
So, as I mentioned, that is for us, you see that two very important enzymes here play a role, and here is a mechanism how the G protein was regulated. It's just a review what we have learned with the G protein to compare with RAS. So the mechanism is very similar for how the protein get activated. Here you can see the alpha subunit when it's bound to GDP is inactive, when it's bound to GTP is activated. And do you remember when signa is in, we have ligand is bound to the G protein couple receptor, then the alpha subunit release GDP. So GTP can bind to the alpha subunit and is activated. And after that, GTP is hydrolyzed into GDP and is become inactivated again. So please, before the next lecture, have a quick look at this slide again, and then we're going I will continue this part. We still have some more slides, and then we talk about tissue after that, okay? So good luck for your midterm exam, okay?
Thank you.
See you next week.
See you.
Bye.
Em cảm ơn ạ, em chào cô.
Have a good lunch.
Bye.